All right, Ken, a member of our Carnivore Certification program, also a member of my early access program in my biochemistry course. Fortunately, congratulations to you on that as well. Good to talk to you. How are you doing?
I'm doing fine. Thanks a lot. And thanks for having me as part of that beta test student
Mhm. Oh,
group of students for your biochemistry class. That's very exciting for me.
yeah, yeah, yeah. No problem at all. No, it's going to be a it's going to be a very fun process. And I'm really looking forward to like really getting it started. I know that there's been a slight delay just because of circumstances, but that is not I have not lost sight of that. So I'm looking forward to that. How have you how in terms of the program that you've already started and you've already done a lot of, if not completed entirely? You'd have to remind me of that if you haven't told me already. How are you enjoying, or once again have enjoyed, the carnivore certification, the nutrition program affiliated with Carnivore Coach Peter, headed by Carnivore Coach Peter?
Oh, I definitely enjoyed it. I have completed all the modules. Um, so I went in thinking that my main motivation is to learn more about the physiology and diet and hormones, vitamins, etc.
Mm-hmm.
Um, and to be honest, I was kind of hoping for a lot more because I was expecting to take several months doing it, but I managed to get through fairly quickly. But that's also a reflection of just how obsessed I become with these things when I get going. So, I mean, it
Yep.
was one of those things where once I started, I really, in all my spare moments, I kept going back to it.
Yep. Yeah, no, I completely understand. That's one of the reasons I'm putting my course together, because Peter's course is designed to be, baseline introductory information so that you can, as quickly as possible, while also being as measured as possible, um, getting people to be able to immediately take on clients because of the huge demand for coaches that actually know what they are talking about, even just a baseline understanding of mechanisms that pertain to diet. So but I wanted to put one that either could be taken on its own before anyone takes that program, which I think everyone should be doing that. That has the ability to become, internationally accredited, which is once again everyone, um, to become a certified nutrition and then also fitness coach. But also, you know, I wanted it to be for people that may have already taken it too, because then you get even deeper. I mean, you're a mathematician, so you already have that mindset. I was someone that was extremely fluent in mathematics in high school, and I wasn't just fluent in it. I really, really, really enjoyed it, and I sought out more complicated um, processes and formulas, um, just to entertain the abstract. I wasn't even trying to turn the abstract into the practical. I was just wanting to solve equations. So that was
Oh,
kind of the
nice.
mindset that I had. I'd make my own equations up at home and spend hours doing them.
Oh,
That's what I
very
did.
nice.
Yeah, that's what I did. So I think that it's a similar mindset, even if it's not the exact same. So that's why I wanted to make that course. But um, fortunately, you're talking to me and you're able to ask me questions now about any more intricacies that you wanted to get into that weren't covered in that course or in that program. So I'm looking forward to taking those. I know that you said they're not in any exact order, but that's totally fine. That's how I write questions out too. So
Okay, well,
yeah.
wonderful. But actually, before I get started with those questions, just what you just said raises an issue that I think about quite a bit, which is this baseline level of knowledge and understanding for a carnivore coach,
Hmm.
because it is definitely a topic which seems enormous. And at the stage I'm at, the more I learn, the more I realize I don't know. And it just seems like, the amount of knowledge that I do not know grows faster than the amount of knowledge that I am learning. And at some
Yep.
point, if you're going to coach, you want to be in a position where you kind of know what you're talking about. And
Hmm.
it's different from mathematics because, in mathematics, you have proof. So when you know something, you know when some idea cannot possibly work to prove something, and know the essence, the key facts that are needed in order to prove something. Um, but diet doesn't feel quite that way. And so, what is this baseline level, and how do you know you've gotten there? And that it's I may never have the confidence to actually coach somebody for real, if you know what I mean.
Yep. Well, okay, so you and I basically we're sharing the same experience, something that I've just recently started to get over, which is imposter syndrome, sort of insecurity in your own beliefs about something. Because exactly as you said, the more that you learn, well, every question that you get answered, that you have, that you start to find the answers to, it opens up about five other questions. So the net result is actually a lack of knowledge, not a gain of it, at least from our perspective.
Exactly.
Um, and so this is why, if you actually go back in my previous videos, when I first started making them and critiquing people, and eventually making monologues and just venting about whatever, you'll actually see that my emphasis on things like epistemology was extremely lacking. So, like the study of how you actually know something to be true, like what is truth in the first place? What does it mean for something to be true? What is objective truth? What is objective reality?
Right.
um, but the reason that that became ever more salient, or salient in the first place to me, was because of just the idea behind, how do we really know any of this to be true? Like, the more you start thinking deeply about anything, something that superficially, from initial perspective, general perspective, is something quite trivial, or something that seems to have a bottom, uh, that is very, very close to the surface, but in reality, it goes way, 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 way farther down.
Yeah.
Um, yeah. So what I'll say from my experience thinking about this stuff 24/7. I mean, seriously, like off camera, that's all I'm thinking about is just, I mean, not necessarily diet, but just like the world. um, I can tell you that from my very, very, very incipient experience, because I'm only 22 years old, that doesn't really, you get comfortable with the ignorance a bit, and knowing that you never become fully complacent in the ignorance. You never stop trying to um, ameliorate the ignorance, so to speak, and fill it with knowledge. Um, but you get comfortable with the imperfections of the models that you construct in your head, or you become aware of, that are already constructed in the universe. Um, so, as much as that might be, depending on who you are, disheartening, um, it's something that I still like entertaining, even if we know that we can't exactly reach a definitive bottom to that pit, if that makes sense.
Yeah, that makes sense. But I feel like I always have to be prepared to have the rug pulled out from underneath me.
Mm-hmm.
Kind of like in Batman stories, suddenly Batman pulls out some gizmo that solves the problem for him, and it's some gizmo that has never been seen before.
Yeah.
And it's just like vitamin C, you might go around thinking this standard idea that you're going to get scurvy if you don't have vitamin C in milligram amounts. And then someone says, but wait a minute, it competes with glucose for the glucose receptor.
Mm-hmm.
And well, that like you never know when lurking around the corner is some piece of knowledge you didn't know that's going to completely change the story.
Uh-huh. And that's also why, when you're looking at logical fallacies, the inductive certainty fallacy is a fallacy, because you can never ever predict, just because you've seen something happen and therefore have accepted it to be true, uh, multiple times without fail, without any deviation away from that result, from an observation, from a chance experiment, whatever it may be. You can't guarantee that in the future, whenever you could perform that same experiment or observe that same phenomenon, that the same result will occur. Never.
Right.
Like, you can I mean, you can believe something's true for hundreds of years. Does that actually make it true? Um, Not necessarily, but then that necessarily gets into a conversation of, like, what do you mean? What does it mean for something to be truthful? What does it mean for something to be true?
Right.
Um, which is the essential, fundamental question of epistemology. And to say, I mean, to pretend like we actually really have an answer to it, I mean, is is a demonstration of, I think, sometimes willful arrogance in a way. Like you kind of
Yeah.
yeah, yeah, like like it's just acceptance based on fear or intimidation. Because you're so intimidated by the fact that you can't find a bottom to the pit that you're in as a human being in the real world, that you must sort of construct a fake one in order to make yourself feel better. Like, yes, we actually do know we do have a definitive answer to that. Like, it's kind of, I don't know. It's something that I've written about before, actually. Maybe I'll write a whole book on that. Who knows? But I'll say that I've written a lot about that because I've been addled by that reality, in as much as what we consider reality is reality.
Yeah.
um, right, like this is why so many scientists, I think, they go into existential crises.
Oh, yes.
Like, there's so many of them, because I mean, what do you do with that? What do you do with that? And I think that I have an answer that has sufficed for me personally, but it may not work for everyone. But yeah, yeah, no, I understand what you're saying. Constantly being insecure to even really, I mean, it is imposter syndrome, isn't it? If you're
It
if
is.
you're if you're afraid to speak upon something because you feel like you are not, I mean, it's a consequence of being a humble person, which is the silver lining. Um, but it's that's what happens whenever you recognize the vast ignorance of all humanity. Like, what we know a lot less than we actually think we know. Like, we live in a post enlightenment era where people think that we know everything. Like, oh, we found the answer. It's all found in science, by the way. And it's like, so many questions arise just from that statement alone. What do you mean by science?
It's true.
Like,
So true.
yeah. So yeah, we'll keep all of that in mind whenever you ask the questions, because I mean, this is why my videos, whenever I make these critique videos, I'll react to like 15 minutes of a video, but it'll take over 2 hours sometimes. Like, this is why, because every single thing that I pause for has like 10 different strains, and if I only cover 8 of them, it's
It's
incomplete.
right.
Like, I can't not say it. So I know it bugs some people, but also, I mean, for people like you, it doesn't, I'm sure, because that's what you think the same way. So
Okay, well, it sounds good. So I
yeah.
guess I'll start with my first question then. And I roughly put them, I think these first questions are just sort of factual pieces of knowledge that I'm missing that I'm hoping you can just quickly fill in.
Hmm.
So my first question is, where do the long chain fatty acids come from that enter the CD36 receptor on a cell membrane? Because I guess I thought that fatty acids would be found inside of lipoproteins. Um,
Yeah.
yeah.
So there's two primary ways that have been observed. You've got two different ways. The first thing that people think of is, of course, whenever you eat the fat. So when you eat the fat, there's a very, very convoluted process, which, fortunately, since you're in the early access program, we'll actually be getting into that, because there's a whole lesson. It's called lipoprotein metabolism, where whenever you consume fat, fat is the most significant stimulus for a compound called cholecystokinin in part of the gastric system. I believe the gastric mucosa, or at least the jejunal mucosa. And what happens is that will stimulate a bile secretion from the liver, and that bile will break apart the fat. It's a long process involving, pancreatic lipase and all that. But the at the end of the day, what happens is when you consume these fats, they get absorbed from the small intestine into lipoproteins called chylomicrons, and those chylomicrons will they have an ApoB 48 receptor, I believe, on the outside, um, as opposed to ApoB 100, I believe, for LDL. You may have heard of ApoB 100.
Yes.
Yeah, yeah, yeah. So ApoB 48 is just a different. It's very similar in its protein structure, but it's a slight differentiation genetically, in the gene that encodes for that protein. But they get administered into the lymphatic system first, and then they enter the bloodstream through the thoracic duct. And what then happens is, according to the I to G ratio, according to the insulin to glucagon ratio, you've got you've got an enzyme called lipoprotein lipase that's present on the interior of the capillary endothelium. And what'll happen is, again, according to the IgG ratio, it'll activate some lipoprotein lipase if it leads to fat cells, for example, and it will actually downregulate the enzyme in the capillaries that lead to muscle cells. I can't remember which is which at the top of my head. If it's higher insulin leads to lipoprotein lipase activation in fat cells as opposed to muscle cells, or if it's the opposite. Either way, what happens is they can either be broken down in fat cells or muscle cells. Usually, if they're broken down in fat cells, it's to be stored until lipolysis ensues again a few hours later, and then they can start being broken down again for use. Or they're broken down in muscle cells for use. Um, and so that's one way that they can be they can be provided to a cell is that lipoprotein lipase will actually break apart the triglycerides in the chylomicrons. Because yes, the fats are broken down in the stomach first, but then they're resynthesized into triglycerides before they're packaged up and then um, provided to other cells of the body. Um, and then the other way is that during lipolysis, the triglycerides stored in fat cells, typically fat cells. I mean, you have fat everywhere, but typically this is in fat cells. They get broken down into their individual fatty acids and also glycerol molecules. So glycerol is water soluble, so it'll immediately just enter the bloodstream and it'll be used for certain processes according to demands by the body. But the fatty acids are obviously not, water soluble, so they need a protein carrier. And this is why we have lipoproteins to carry things like cholesterol and other fats. But whenever they're being broken down from fat cells, lipoproteins don't carry the fats. It's actually albumin in blood plasma. They actually carry them directly without the use of an actual sort of encapsulator, I guess you could say, sort of a dome. They actually just carry them. Um, I don't really know what word to use, but they're more free floating if you could think of it that way. And what is referred to as non-esterified fatty acids. These are NEFAs. You'll actually see abbreviated. And then what'll happen is they can be taken up. Usually, if it's in the case of lipolysis, you've already got a low IgG ratio, so they're taken up by muscle cells. They're taken up by liver cells and also the heart. Those are the primary tissues that engage in beta oxidation. So those are the two ways that cells can yield um, fatty acids. Through the diet, but also through lipolysis whenever you're not eating. Or if you're in a ketogenic state, sometimes lipolysis is actually still occurring even after you've eaten food. If after you eat the food, you actually get a decrease in the IgG ratio, which happens in some people, which I've covered, in other videos before as well. I think in a few of them.
Mhm.
So
And is the CD36 receptor involved in both of these methods?
yes,
Okay.
yeah. The administration of fatty acids is through CD36. You've got some exceptions. For example, short chain fatty acids, they seem to be able to permeate without CD36. They're also, um, you know, again, since you're in the early access program, you've seen the drafted beta oxidation lesson, at least part one, and you see how these fatty acids primarily, generally, they're what's considered carnitine dependent. So they actually have to go through CPT1 and CPT2 to get into the mitochondrial matrix. Short chain fatty acids, I believe, completely bypass that process. They're definitely not carnitine dependent. They're non-carnitine dependent, but they may still enter through a transporter, not simply through, extremely passive diffusion. Um, I can't remember at the top of my head, but so there's slight nuances there, but generally speaking, the prototypical fat that you're going to be using is a long chain fatty acid, and so those are carnitine dependent, but also they are CD36 dependent. They enter through CD36. Yeah.
Okay. Great. Thanks for that.
Mhm. By the way, actually, you know what? Before you get into your next question, because you're into the intricacies, you might there's a lot of people that not a lot of people, but some people will actually ask, how do we actually know this to be true? Like, how has this actually been observed? Um, you may have heard me mention nuclear magnetic resonance spectroscopy before. It's a really long name, but really what you do is it's basically a machine, to simplify, that can actually shoot photons out, and they reflect back. And so you can actually observe imaging at a resolution of one photon per 1 millionth of a second. And you
Wow.
can actually see these things happen, sort of the steps, in an organism, live in a way, like actually after observing the image, yes, and deducing from that what's going on. But that's how we, of course it's repeatable in multiple people, and that's to be expected, because as much as we're genetically different, all of us, we still are extremely genetically similar, or else we wouldn't be the same species. So things, fundamental processes like oxidative phosphorylation, uh, lipid transport, those are going to hold constant for human beings across the board. Fortunately, of course, though, it has been verified through actual observation in multiple individuals without fail. So that's the typical way that we observe these things. Same thing with muscle energetics. All of these processes are verified and validated to be true through these imaging techniques.
Wow. Neat.
Yeah, yeah, yeah.
Well, okay.
Mm-hmm.
Um, so my next question is, how does the body remove arterial plaques, and how quickly or slowly does that process take?
Yeah, so in terms of, usually when we're talking about atherosclerotic plaque, we're talking about soft plaque first, because then it takes a while to become calcified. The calcified plaque, it doesn't really seem like the body does too well with breaking it down the same way it does with soft plaque. With soft plaque, what happens is you have macrophages, a type of white blood cell that eat things up. And what you have is, you've got macrophages that come along, and they take up a lot of the foreign, or what's considered foreign by the body, if we're going to slightly anthropomorphize it a bit. um, it tries to take up things like, it does take up LDL particles that are in the plaque that are either oxidized, glycated, whatever it may be, and other compounds that may be deranged because they've been there for so long. Um, because the damage that they're there for has not actually been ameliorated in any way, or at least enough of a degree to warrant them having done their job, having completed their job. So macrophages they compose a lot of the plaque. The so that's we can infer from that that's one of the ways in which the body is trying to take up the things that make up the plaque. It's macrophages taking up these foreign proteins. However, that only happens whenever these proteins are deranged. It doesn't happen whenever they're not deranged, because, well, if that were the case, then you'd have, I think that you'd see a lot more atherosclerosis. Um, and it would also take less time to accumulate. Um, but in the cases where these proteins are not um, deranged in any way, they're just there for longer periods of time. They haven't become oxidized. They haven't become glycated. The body isn't actively trying to get rid of the plaque. It's actually trying to form it. That's what the LDL is there for. The LDL is there to supply the tissues that are damaged with cholesterol, because cholesterol is one of the fundamental elements of a cell. It forms the cell membrane. It forms your lipid rafts. It doesn't even just help to separate the phospholipids in the phospholipid bilayer of your cell membranes, but it actually forms lipid rafts, which is, sort of larger concentrations of cholesterol that hold, that sort of integrate elements of the cell membrane together. Um, and so they start to build. But what's supposed to happen, though, is the injury is supposed to be repaired, and then the LDL can sort of de-sequestrate itself. I don't know, de-deposit itself, and sort of take off, and then go back to the liver to be picked up. Of course, this all implies volition for sake of conception, for ease of conception, but in reality, it's all chemical pulling, chemical pushing, chemical pressures. That's what's supposed to happen. But yes, if those proteins do not leave and they become deranged, then the body is actively, it's trying to take those proteins up, and so you'll see macrophages in atherosclerotic plaque but that actually in turn builds plaque. So I guess the body isn't really ever trying to get rid of plaque. It's actually trying to heal the injury as much as possible, which as a consequence builds the plaque up. But what's supposed to happen is naturally the injury is supposed to heal, and then as a consequence of that, the plaque, there's no pressure for that plaque to be there, so it starts to regress. Think of the best way that I can put it, just to put a final note here, is they're actually referred to as microcalluses in certain papers when you read them. Just think of them as calluses. Like if you abrade the skin enough, I mean, I actually have one on my thumb here for putting a bunch of push pins into the wall. Um, you'll build a callus because there's abrasions going on and it's damage to skin. It may not be a super serious injury, but it's an irritation nevertheless, and you build up a callus. But once you stop abrading the area, there's no pressure for that callus to be there, and so then it starts to regress because that energy that is being used there, according to the Darwinian theory of evolution, it's there's no pressure for it to be there, and that energy could be used elsewhere that the body deems to be more important, so it can take it away and allocate it elsewhere, even if it's a small callus. We're talking about a bigger callus and a more significant one when it comes to the interior of the endothelial of the endothelium.
Right. So I guess I'm specifically wondering what the mechanism is behind this receding of the plaque, and the time scale in which it takes place. Because I gathered that some people feel that problems arise when glycation damage or inflammation occurs at the same place in an artery with too great a frequency, that before a plaque can recede, another one is formed on top of that, and another one is formed on top
Mm,
of that. And then you
yeah,
get, yeah. So
yeah,
how
yeah, yeah.
much time do you have? How much damage, how much do you have to limit that damage, the frequency of the damage, in order for your body to be able to handle the plaque load?
That's a good question. I think it's going to vary depending on the person. Um, I think that, yeah, you bring up a good point because it does become a positive feedback loop. There's initial damage, and then you just keep damaging the tissue, but then it doesn't even just become more damage to tissue. Then it becomes a derangement of the already existing plaque in a way, which actually leads to more plaque because you've glycated the elements of the plaque and you've oxidized the elements of the plaque, and then you build more, and then you build even more, and then, um, so I guess that there's not a definitive answer that I can give on that because I mean, in terms of trying to determine a certain frequency by which the plaque, a certain limited frequency by which the plaque can actually start to regress. I know that I just said that it's highly individual, and I'm not saying otherwise. However, I think if you're going to look at the dispersion around, if you could actually collect data on that and the dispersion of the frequency, mean plus or minus the standard deviation of the frequency by which you actually observe plaque to be regressing, assuming causal relationship there, I think that the dispersion would actually be a lot lower than other random variables.
Okay.
Despite, but I can't give a definitive mean for that. I'm not really sure. I don't know if there's even really a way to measure that definitively. I'd have to put something together hypothetically and then tell people to do a study on that or something because I can't do the study myself. But at the
Yeah, and
moment,
just
yeah,
anecdotally from some of those interviewees of Dave Mack, for example, they do speak about like their CAC scores going down. And I was curious about the timescale for that to occur, and what the circumstances were for that to happen.
yeah. Well, here's another thing too. A lot of I'm not when I say a lot of people, I don't know the exact empirics because I don't think anyone does. But I've heard people say that whenever they go carnivore, they'll know their CAC score initially, and it's quite high sometimes, like pretty high. Which we know it only measures calcified plaque, and so it's not the most unstable plaque. The most unstable plaque is the soft plaque. But nevertheless, it means that there's been plaque there long enough for it to become calcified, and a lot of it to become calcified. what they'll see though is sometimes even after a year of being carnivore, their CAC score before it comes down, it'll actually go up quite a bit.
Mm.
And that was interesting when I first saw it because I mean, my initial thought was, wow, so they're still getting damage to their arteries. Like, what's going on? So they must be doing something wrong. Like, their body might not be adapting to it appropriately. Like, who's to say? But then I thought further. I was like, okay, well, maybe, but calcified a CC score, if it measures calcified plaque, that means that they could just instead of having a an increase in the amount of plaque buildup, they could theoretically they could have the same amount of plaque, but more of it is just calcified. There's who's to say that they're actually just building
Ah,
more and more plaque? What if a lot of it is just becoming calcified now that wasn't observed on the CAC score before because it existed as soft plaque? You know, they didn't actually measure non-calcified plaque volume, NCPV,
mm,
which is a newer phenomenon that I'm seeing, or at least it's become more mainstream if it existed before. Um, but that's also something that I've seen sometimes when I mean, in terms of the frequency by which someone can see a regression, the frequency of damage, like we were just discussing, that you would have to reach, you know, the decreased frequency by which you can actually expect a decrease, a regression of plaque. Sometimes, I mean, who's to say that they're not exhibiting a plaque decrease in terms of soft plaque, even if they have an increase in CAC score? So then it becomes even more noisy. Like we
yeah.
need like, so I mean, I never want to like I'm a curious thinker, so I'm not using that as some sort of like scapegoat for people exhibiting a higher CAC score when going carnivore. It could very well be because of exactly what people are saying, like their people's initial thoughts, like, oh, this is actually getting worse, not better, and then we have to actually look at what's going on. But I do think that it's if you want a better assessment of that, you'd look at non-calcified plaque volume. Um, I'm just I I'm I worry about the expense of that compared to a CAC score at the moment if it's quite nascent. And then same thing with the potential radiation exposure.
Yeah.
I'm not familiar
Right.
with that. Um, so that as well though. Pretty sure there actually is radiation exposure with that.
Yeah.
So you want to be careful with that.
Yeah, for
So yeah,
sure.
there's no definitive answer I can give for frequency. I just from anecdotes. I mean, I've heard of people saying that they can see a decrease in CAC after a year. I've heard of that before, but who's to say, really, for every single person? It's really difficult to do that.
Okay. So my next question is, and these are so random. I'm sorry, I'm jumping from one
Good.
arena
No,
to
no, no,
another.
no. They're good. It's good.
Um, so is the concept of optimal levels of nutrients and vitamins meaningful in the sense that, you know, how when you go to have your blood drawn, the doctor reports on the results, they say, oh, your vitamin D is in the optimal range, your vitamin C is in the optimal range, et cetera, et cetera. Vitamin B12 is optimal, or they say you have a deficiency. To what extent are these meaningful? Because like I think I understand the situation with vitamin C, but what
Hmm.
about for other vitamins? And are they mostly like vitamin C, or are they mostly like there is kind of a optimal range that has real practical import in health?
Yeah, so I think the best way of answering that question is to actually assess how they even come to formulate all of these normative ranges.
Mhm.
Because when we talk about optimal ranges, it's in reference to the normative ranges. Typically, what they'll do, because this is what they have to do. Let's say that you're a scientist, you want to assess the optimal, the normative range of a certain vitamin. Let's just say, for example, vitamin D, and you go, okay, let's try and find this out. Well, you know at the top of your head, you know immediately. Well, I can't measure every single person in my country. I can't measure every single person in the world. Um, at all. And I definitely, I mean, I can't do it in my country either, my respective country. So what I will have to do is I will have to collect data that is already being collected regarding this topic. So I'd have to refer to levels that are actually calculated and yielded after measurement. So what you do is you try and find data, you try and access data from every clinic that you can across your country. That's typically what you're doing. You're trying to form a large data matrix, large data set. And then from that, what you do is you perform a simple random sample, at least ideally. You assess of course, actually, you would really want to look at a probability density function and look at the shape of that, um, to assess the distribution of that as well. But ideally, yeah, let's just assume it was normally distributed. Then you do a simple random sample, and then from that you'll calculate the average level or the median level, plus or minus whatever, and then you'll use the plus or minus figure in the form of a standard deviation or a median average deviation, and you'll form an interval range that way by which you're considered to be within normal ranges. That's really what they're doing.
Mhm.
Um, in reality, side note, they're actually not forming an interval range that way. They're using a confidence interval. So they're actually using inferential statistics, which makes it even worse, because that's not. I can go on and on about confidence intervals, and we can get into it if you'd like. I enjoy talking about that stuff so that people understand it, but it's a very dense topic. But let's just give them credit. Let's actually give them credit. Let's say that they weren't even doing that. Let's just say they did exactly what I said they should be doing from their best available tools. So using descriptive statistics. Well, then you must ask yourself, okay, well, they took this data. Where did this data come from? Well, it came from clinics. Okay. Well, these clinics, they are taking people's blood, and that's great. That's how you access the data. But whose people's blood? Are they representative of the overall population that you're trying to extrapolate the values to? Well, who goes to the doctor's office?
Right.
Healthy people do go to the doctor's office. I mean, I know I have family members that even though they're healthy in terms of presentation, they feel great, or at least they think they do. That's a discussion for another day. But they they report to be feeling good and fine, but they go to their yearly checkup just because they think that's a routine checkup. Like, let's just see if I'm okay. Let's see if let me get screened for cancer, this and that. Some people do that. That's fine. But is it the vast majority? Well, I could tell you right now from my experience, no. And from everyone around me, the majority of people around me, no. They go to the doctor whenever there's a problem. They go to the doctor whenever there's a problem. They're sick. They got injured, but it's not severe enough to go to the hospital. Um, so you're referring to a demographic of people that is overwhelmingly um, aberrant in their presentation. And so you have to be, I mean, so to get back to your question, the reason I said that the best way to answer that is to assess how they actually come to these normative ranges. It's like, well, if they come to the normative ranges that way, then there's two things that you're left to realize, that you must realize. And the way to realize them is actually by formulating them in terms of questions. Can you be within those ranges not really being representative of healthy people, healthy being determined by presentation and actually lack of lack of disease process in the first place, but especially particularly given our confines, identifiable disease process. Um, can you be healthy within and be within that range that isn't reflective of healthy people, isn't representative of healthy people? Yes. Can you be unhealthy and represent and be within those ranges? Yes. Can you be healthy and outside of those ranges? Yes. Can you be unhealthy and outside of those ranges? Yes. So this is why, this is usually one of the things I'll say oftentimes is we need to zoom out. Like, we really need to zoom out, because getting into the normative ranges and looking at whether you can, fit within this, and whether it's utilitarian at all to even observe these, it's getting into the weeds. And sometimes getting into the weeds is fine because there's not a bunch of noise around that. But a lot of times, most of the time, in my experience, there's way too much noise. We need to zoom out, like see the forest for the trees. What is the one of the most archaic principles in all of medicine? It's the Hippocratic Oath that says do no harm. It's very vague what that means, and people treat that as like, don't do any direct harm. But that's if that were the case, I don't think Hippocrates would have had to make that statement, because everyone knows not to literally inflict harm upon your patients. um, what it really means is something deeper. It's a broader statement that encompasses a lot of different principles. The primary one, the most relevant one to the topics that I talk about, are is if your patient presents to you with no symptoms and you cannot identify a disease process, even if you believe there to be a problem, you must not intervene in those circumstances. Are they presenting with symptoms? Okay, you've done your blood checks. They're way out of whack in their ranges, in their the normative ranges. Okay, look way out of whack. All right, but there's no identifiable disease process, and there's no symptoms being exhibited by the person. And perhaps, in the case of many carnivores experiences, they feel the best that they've ever had, that they ever have. Well, that is a situation whereby you should not really intervene. And maybe even if that's true, you could say, well, maybe you should still be concerned. Well, that's an opinion that you can keep to yourself, and I mean that in not a, I don't mean that in a snarky way. I mean that on an ethical basis. I mean, like I have opinions like that sometimes, but it's my responsibility to keep that to myself because there's no evidence to suggest that they are actually exhibiting a problem, like that I should intervene.
Mhm.
So yes, in terms of whether they actually have utility or not, for overall blood tests, I would refer to blood tests only if the person is presenting with symptoms. Then you can actually start formulating, you can intervene and start formulating hypotheses based on those blood markers. Um, Outside of that context, formulate opinions, that's fine, but understand that there are vast limitations. The limitations of science are oftentimes, if not always, more important than what science can actually demonstrate. So that's typically that's where I'll leave that, if that makes sense.
Oh, yes. But so, I mean, I understand there are problems with the way these optimal levels are determined. And I suppose if they go back 100,000 years and test vitamin C levels in a healthy population of humans, then they might have gotten a completely different range from what they're
Mm-hmm.
getting today. But I was actually trying to get more at whether you think the concept of optimal levels is a sound one,
Mm.
or is that just a silly thing to try to even determine?
Yeah, I'm understanding your question more. Um, yes, typically, yeah, the concept itself, I suppose, can be of utility so long as the population that is actually being observed, or the set of populations, because you want to, science is one of the principles of science is reproducibility of results. Um, so long as the population is living as indicated of a lifestyle as possible according to the prevailing scientific theory, because that's the best that we've got, the Darwinian theory of evolution and what we evolved most to withstand genotypically across our genome. Um, the problem is that there's two problems. Number one, no one's doing that, it seems. And second of all, as a consequence of the first problem, every population that you study is doing something, is eating a completely different way than the other, actually. So it's not even that like every population is eating the same thing, it's just not appropriate for our species, ostensibly, according to that theory. It's that every population you're studying or you're observing, is they're eating completely differently from each other oftentimes.
Mhm.
Um, so that's the problem with even getting, if you're going to say that there's utility in the concept of optimal ranges, those optimal ranges. Um, but even if we were to do that, I think what's best is is just assessing. Okay, again, your presentation and any identifiable disease processes, and then yes, we can have those optimal ranges as a stronger inference of a problem if you're presenting with symptoms than we actually have today. So yes, the concept does have utility. I'm not going to pretend otherwise, but I think that to try and to try and focus strictly on one thing, not to say that you're saying that we should, but um, everything is a composite. It should be, every conclusion we arrive at should be through composite inferences, instead of just like normative ranges or, so sometimes you can use one thing, like again, identifiable disease process. Okay, we need to intervene. Like, okay, so we've observed one thing. We don't even need to look at anything else. Let's intervene. But before we intervene, then we can look at other things. But to determine whether we should intervene, there are things that you can just use one of. But most of the time, you're going to want to look at composite inferences. So yes, the concept can have utility, but definitely not in today's world. I don't think. Um, Maybe not in my lifetime, our
Okay.
lifetime. So
Yeah.
yeah,
Got it. Oh, thanks for that. Okay.
yeah, yeah.
So next, for any particular nutrient, is there a general theory pertaining to whether or not that nutrient can effectively be obtained through our diet? Or is this the sort of thing that has to be tested individually for each nutrient and each food item that contains it? And you just have to have some people eat that and see whether it gets through or not. So,
Oh, in terms of absorption,
yeah, for example, like CoQ10, is that an effective
yeah,
dietary supplement? And then,
yeah. The okay, so again with nuclear magnetic resonance, a lot of times what you can do is you can assess the digestion of a a nutrient and the amount that is, let's just put it this way. Usually that's not done. What's actually done, because it's less expensive, is you'll assess it through indirect means. You'll look at the absolute amount of a vitamin, a mineral, whatever it may be. You'll have a set of people consume that vitamin or mineral, and then what they'll then observe is, okay, how much was absorbed into the blood postprandially, so after they consumed it,
mhm,
and then so they just look at the absolute increase in the blood level, right? And then if you're studying a group of people, then you'd have to observe the mean increase, right? So because then you're looking at multiple people, and then the dispersion around that increase, um, and then the reproducibility of it on other people, and then all that statistical stuff. But either way, you'll look at that, and then you'll compare that not simply to the absolute amount that was in the unit consumed, but also how much is actually excreted. You can sometimes measure that. They do that with protein, sort of on a more macro level, because protein's a macronutrient. Um, they'll measure the amount of nitrogen containing catabolic byproducts, because amino acids made up of nitrogen and all that are contained in urine and fecal matter, and then compare that to the amount of nitrogen consumed as protein within that same time period. There are confounding variables. To pretend otherwise is silly, but I think that's their best inference, because I mean the confounding variables are like, okay, well, how much is the absorption dependent on your activity that day? Well, for protein especially, it's going to be very dependent,
ah,
because
okay.
so there's always going to be confounders. But for vitamins and minerals, there's also going to be those same confounders. Like, what was your demand for that mineral that day? What was your demand for that vitamin that day? What happens when you consume it with this food versus this food versus this food? That's another thing. You got to test things in combination with one another. It's the same reason why on drug labels or on bottles, um, you'll see contraindications, and they'll tell you what not to mix it with, because depending on it affects absorption, it can influence its function in the body when absorbed. So the best way, again, you're on the right track, because if it's a very similar answer I'm going to give here that I just did to the last one. It does have
Exactly.
utility to
Ah,
measure absorption. It does have utility, like to measure that absorption on a clinical level, and to assess the repeatability of that. But at the end of the day, what you're going to have to do is, okay, well, eating this food, when these people eat exclusively this food, what is the incidence rate of a deficiency in said mineral or vitamin that can actually be directly tied to a deficiency in that mineral or vitamin? They're called deficiency diseases. We've done this with fiber, for example. There are no deficiency diseases that are observed in human beings, on any human being studied, and for the duration of time that they've been studied. Again, I always try and convey the limitations in the explanation
mhm.
that sh- that, that are correlated with an abstention of fiber. There's none observed. Now, you always want to be careful. This is why it's important to know the logical fallacies. You don't want to make appeal to ignorance fallacies. Well, since there's no evidence of that, then it must be true that. No, but what you want to say is, if there is no evidence, then it's merely speculative. Provide some more evidence of that. From my experience, I've never seen people have deficiency diseases either. And frankly, it's actually quite the opposite. They report feeling better than they ever have, digestively speaking, without consuming fiber. So there you go. So that's kind of how it's always a broader inference that should be made, I think, than a more granular one. As much as granular ones can have utility, particularly, especially with things that have not even been ubiquitously consumed yet. So, for example, when you're drug testing, like absorption rate, because you can't just administer the drug into the public first, and then in order to observe the incidence of absorption problems with the drug, you actually have to assess it clinically first before you can get it passed. You have to do these studies first. So yes, it does have utility, particularly in that respect, but also yes in things of like overall nutritional inputs. But should that be what's actually assessed? Well, I don't think so, because this is one of the reasons why this is one of the reasons why when people talk about bioavailability, it's important and it's a good inference on its own. Like, okay, things that are more indicated for our physiology tend to be more bioavailable, and it makes sense why that would be the case. But you don't want to do a reverse causality kind of thing, which means that if something is bioavailable, then that means it's more good for you or more beneficial. I see that a lot in the space, and I'm not really a fan of that.
Mhm.
You may have seen that before. Like, oh, well, this is the most, it's good for you because it's the most bioavailable thing. I'm like, well, that's not why. Because then literally anyone can turn around and say, well, this thing's actually more bioavailable than the meat and eggs. Therefore, that's more healthy. It's like,
Right.
I mean, that's not that's not logical. So that's why I always say, okay, well, we can pretty much say that something's not indicated to be relying on if it will necessarily lead to nutritional deficiencies. Okay, because of course that's not appropriate for an organism to be exhibiting, right? So, do we have strong enough inferences that a carnivore diet, for example, overall, the nutritional inputs on a carnivore diet that is most congruous with what we ate as a species from every inference we have, leads to nutritional deficiencies, or at least is associated with nutritional deficiencies? No, we don't. Because then, and if there are any studies that purport to show that, then you must ask, how was that actually how was that characterized? Nutritional deficiencies. Well, they exhibited less than the normative range. Yeah, well,
Right.
then we're done, because that's not how, like, actual deficiency diseases. Did they present with diseases that were in genetically normal people that were directly correlated with the nutritional inputs of a carnivore diet? There has been nothing to suggest that. Not to say that there never will be. Again, appeal to ignorance fallacy is a fallacy. However, that means that it is merely speculative, and I highly doubt that that will ever be seen. So yes, I haven't lost sight of the question. Just to emphasize once more, no, it does have utility, yes, but overall, I think that we should be looking at the broader picture, which is what are the incidence rates of deficiency diseases according to the nutritional inputs of these people, and that's really your best inference that you're going to get.
Mhm. It's funny how this ties into what we are discussing at the outset too, because I totally get your point about the broader, the better the context that you understand for this individual, and you're
Mm-hmm.
trying to evaluate. They're presenting with some sort of symptom that's associated with a dietary deficiency in some nutrient, and do you conclude that, oh, you need to eat more of this? And you have to take into as much as account as much as you can about their overall profile, and but then you have to also live under the circumstance that you may be missing things still yet.
Mm-hmm. Yeah.
It's
Yeah. Yeah. Science is it's a lot, like I always say, it's a lot more ambiguous than people think, because there's limitations with every single bit of this. Like, I understand the desire to be more granular with things, because immediately you think that you're getting more fundamental, right? When oftentimes it's actually paradoxically you're getting less fundamental, because of the amount of noise you're introducing into the signal. But like these cellular biological studies, oh, we identified a mechanism that suggests that this compound found in this food can activate this, which can then go on to activate this, which can then cause potentially cancer in mice or something like that. And it's like, wow, I mean, that really means nothing, does it? But that'll hit headlines, man. That
oh,
will.
I see. Yeah.
It does all the time. And I understand the desire to get like that, because when you get to the biochemistry of things, you think that you're getting fundamental. And depending on what you're looking at, you are. My course that I'm going to be building with your help, and everyone else is in that cohort, that very small cohort, is designed to cover the fundamentals of biochemistry, things that are on a broader scale, always to a massive, invariable degree observed.
Mm.
That's the difference. We're not drawing things from fringe pathways and saying, this is why you shouldn't eat this. This is why you shouldn't eat that. No, no. We're learning what has been observed in a routine, regular, reproducible fashion in our genetics, like oxidative phosphorylation, how the mitochondria function. That's not being reductionist. That's like we breathe oxygen for a reason. Like, okay, this is how oxygen is used. This is how we observe it to be used. Um, Gluconeogenesis, like the fundamental pathways are what's important to observe. It's not, it becomes pseudoscience whenever you start doing these cell biology studies where, yes, you've identified a pathway and it's true that like this is what is observed, this pathway exists, this molecule travels through it, whatever, but trying to draw implications from a very tiny fringe pathway that is one gear in a gearbox of millions of gears that are working together in a massive nexus.
Yeah.
It's completely irresponsible. But if you went into that gearbox and you found the fundamental gear of the gearbox, you could make better assumptions. Like, okay, an example would be, like I just said, the mitochondria, oxygen, right? Like, okay, well, if you remove oxygen from the system, we know the whole thing will fail. That's not the same thing as looking at like one part of a branch and saying, I know everything about the tree. That's looking at the seed that the tree was grown from and saying, well, if the tree doesn't exist, well, then this is, well, if the seed doesn't exist, well, then the tree will never exist. The branches will never exist. That's different. I think, do you understand what I'm saying?
I think so. Yes, I do.
Yeah. Like that's what we're going to be doing in this course because you always have to be careful when you get that granular with the introduction of noise, massive amounts. We're not talking about a small amount. We're talking about massive amounts whenever you get into granular details like that. But then the other problem is that you've got problems on a broader scale. It's like, well, if we observe okay, fine. So we can't look at like just the absorption rates on a clinical level because that doesn't necessarily reflect the broader picture. But if we go broader to try and assess, okay, deficiency diseases, the incidence rate of deficiency diseases in a population that is eating a carnivore diet, well, then you've also got a lot of noise too, because, you've got what else are these people doing that you can't observe? What else are these people not doing?
Right.
But it's like, yeah, there's always going to be noise there. You got to pick the inference that has the least amount. The broader picture usually has the least amount, unless you go too broad and then it has too much again. Like, it's like, science is a really, really, really delicate game. Really delicate. I don't think we have scientists that respect the nuance anymore, though, to actually, be delicate with their inferences or their constructed methodologies for studies and investigation. I think people think too linearly, including scientists. X goes to Y goes to Z, instead of realizing that everything is like a clock tower or a spider web, where every intersection on that spider web represents a variable, X, Y, and Z, and every line, A and B, and every line that goes through each intersection is bidirectional. If they
Yeah.
realized that, then I think they'd be a lot more careful and maybe put a lot less faith in observational science like that. There's different forms of science and fields of science. There's physics and all that. That has a lot more. I mean, we're talking through a computer screen right now. Science is important, right? But there's different forms of science, and some deserve more faith and emphasis than others when it comes to purveying the truth of the universe. And I think that this type of science is one of them, if not the main one. So,
Yeah, and definitely a lot of journalists do this oversimplification of the science and
mm-hmm.
convey a very simplistic picture of the situation, even regarding physics.
Yes,
Yeah.
yes, it'll happen with everything. I mean, everyone wants to trust. You grab a newspaper. I mean, nowadays you don't do that, but you read an article or something, and you believe basically everything that's said there. At least this is the tradition until like the last 10, 20 years. Um, Maybe the 70s too with the Nixon thing, but you read the paper and you believe like every article, and then there's one article that's written about something that you know really well, and you find holes in like all of it. And you're like, that wasn't the best thing, but then you still believe but you believe that there's no holes in the other stuff that you don't know. It's like, no, that's what it is with everything.
Yeah.
That's how it is with everything. Like, I'm
Yeah.
sure if they wrote an article about some mathematical some new theorem that explained the universe better or an aspect of it better, and they tried to explain it, I'm sure that you'd find holes in that in the explanations if you haven't done that already. I mean, usually math isn't talked about in the mainstream news at least, but I'm sure that would happen.
Yeah, and
So who's to say that it won't happen with everything else? And it does.
it's
So
true. But
yeah.
I've definitely read papers like epidemiological papers where the authors are actually quite careful pointing out the flaws in the
Mm-hmm.
paper. But then the newspaper report of it turns it into a fact. Author
Yep.
turns their conclusions to these facts.
Yep. I just did a video on that actually, and it's releasing next week at
Oh,
the time of
awesome.
recording. Yeah,
Oh, cool.
it's about caffeine consumption.
Oh, great. Oh, I can't wait.
Yeah. And they say they cite the authors saying we have to be very careful not to draw a causal relationship from this. Um, but I guess the difference in this article was that they actually went on to quote him again, where he says we have to be very careful not to draw causal relationships from this association because there's many possibilities. This could have caused this. This could have caused this. But what we do know is that caffeine was still associated with decreased risk of whatever it was. And I go, no, no, no. So you just undermined what you just said.
Right.
Like you're undermining what you're saying. We have to be very careful not to assert causality. However, caffeine did cause an increase in this. It's like that's basically what you just said. It's like no. Anyway, yeah, it's all over the place because that's what gets clicks. That's really what gets
Yes.
clicks.
Yeah.
So yeah.
Okay, so um, still got more questions than I've asked so far. I don't know how much time you have, but
I have plenty of time. I have
okay.
plenty of time. Don't worry about that. Yeah.
Um, so I've heard some researchers like Thomas Seyfried, they speak about
uh,
therapeutic levels of glucose ketone index. And I was just very interested
okay.
in your take on this glucose ketone index, and because personally, I've never gotten close to the therapeutic levels. And, um, is it something to strive for? And like, do you have to necessarily do extended fasting to get there?
Um. So whenever I hear an index, I'm immediately thinking of a ratio. So I'm immediately thinking this is basically an adjacent constructed ratio or quotient,
Mhm.
depending on how it's presented, to the I to G ratio.
Mhm.
Because if you're going to have higher glucose, you're typically going to have higher insulin because that's going to cause the pancreas to secrete more insulin if the glucose levels are high, as compared to ketones, because ketones are produced through ketogenesis, which is most highly influenced in their production or in the activity by glucagon. So it's basically just I to G ratio, just in a different form, glucose to ketone index, or And so really we can reformulate that question into, well, is there a therapeutic level of an I to G ratio effectively? Which again, I don't think there's a really hard way, a definitive way of assessing that. There's going to be an interval range if there is to be one, because not every single person is the exact same, as much as we are genetically identical, like 99.99% of the way, but
Mhm.
because we're the same species. Um, it is true that if you're someone that has cancer, we know that Thomas Seyfried, he's become very popular for spreading, promulgating the message of glucose dependence in cancerous cells for clear mechanistic reasons.
Mhm.
Again, that deal with the broader elements, so it's pretty sound explanations. Mitochondrial dysfunction, therefore not being able to use oxygen too well. Fats require oxygen because they require to be in the mitochondria to even be used in the first place. Glucose doesn't. There you go. Like, okay, but but also glutamine.
Right.
Um, because there's a way that it can be used in the Krebs cycle still, in the presence of the oxygen that is still present in mitochondria in cancer cells, because they still do respire. Um, it's not like they don't use any oxygen; they just use less, a lot less. Um, but how do I say this? I think I've already said it. It's hard to say that there's a therapeutic level. I think the best thing to do is to trust the broader process, which is cut the carbohydrates. And if you're still having cancer that is either remaining the same or it's getting worse, then you're going to have to get on some glutamine inhibitors, I think, something that interferes with the utilization, at least, of glutamine in the mitochondria. I'm averse to completely writing off Western medicine.
Mhm.
Um, very averse to it because, as I typically say, if you're exhibiting an extreme deviation away from what we evolved to um, express genetically, phenotypically speaking, then you may very well expect it to get so bad that you need another deviation, an externality that deviates away from what we were used to get back to a normal equilibrium, counteract. Yeah, in terms of assessing an exact therapeutic ratio, I don't know if that's really worth doing it. I think that, oh yeah, two negatives can equal a positive.
Oh
That's what I
yeah,
was
and
saying.
the deviation, and I think Professor Seifert himself does not say just do this because I think his patients still undergo radiation to get to this therapeutic level, and then they still do the radiation,
Mm-hmm.
at least in some cases.
Yep. And well, this would also explain why certain ketogenic therapies actually don't work. It would explain that, if glutamine is still a fuel source being used by cancer cells, or if you're, I mean, there's so much, there's a lot of mystery still with respect to cancer. And I mainly think that this is because a lot of people are doing the wrong studies. I think what they should really be doing, like now that we know that cancerous cells use glucose and glutamine, well, now we should look. Okay, in the cases where ketogenic metabolic therapies are not associated with any regression in cancer or an amelioration of cancer, anything at all. There's no improvement over a significant duration of time, whatever that duration of time is deemed to be.
Mhm.
We should be looking at, okay, well, how much do cancer cells seem to influence the rate of glucose production when they're not consuming it?
Mm.
Like, I mean, we can produce our own glucose, but if there's a cell in need of glucose, what can the cancer cells send cellular signaling that can be observed that directly stimulate gluconeogenesis in an upregulated fashion to provide more glucose to the tissue? Because if that's the case, then it actually has nothing to do with glutamine in that situation, or fallacy of the single cause. Maybe it does, and they're not mutually exclusive. It can have both things can be playing a role. Like you're producing more glucose, and what like I want to see studies like that so that we can determine, okay, well, is Western medicine actually still something that we should employ in these situations?
Mm.
Like, I do believe that there's a place for all of that. I just think that the place it's not for 90% of things. It's like for 10% of things, but it's used for like 90% of things. Um, and it's the first employed, you know, um, treatment method. Um, but I want studies like that to be done, and like how much in someone that isn't consuming glucose is glutamine actually used? Can you observe that? Can you I want to actually I don't know if those studies can be done. Um, Money is a big part of this in terms of how much money would need to be spent to conduct these studies. Money is always a problem with the conduction of studies, whether it be because a singular study is a really massive one, or because of the amount of studies that need to be done. So whether it's due to quantity or quality or both. Um, but then we could get a better idea of when it would be appropriate to really implement and employ some Western medicinal practices besides just immediately jumping to, okay, just to be safe, let's put a bunch of expose you to a bunch of radiation. Like, not to be cynical and flippant,
Mm.
but that is what's going on. So it would be really good if we could avoid that
Mm.
to the greatest degree possible. Um, but yeah, more. I always get tangential, but more to the point. Yeah, in terms of the therapeutic ratio, I guess the best way of assessing that, if you're going to try and formulate one, is again get enough of a get a statistic get a big enough sample that provides adequate statistical power for what you're looking for, that can be repeated multiple times with a strong aggregate signal to noise ratio as to the associated what indexes glucose to ketone or ketone to glucose, whatever, is associated the most with regression or amelioration of cancer, while also being careful not to draw causal relationships, also being careful not to extrapolate that to every single type of cancer. That's also important because different cancer cells, they always have the same common foundation: mitochondrial derangement, slowed mitochondrial function, sort of a thermodynamic inhibition of electron transport chain in the mitochondria, which is also really interesting. I reviewed that a few months ago. They always have the same common denominator, or denominators, but they still, some of them actually, I've seen that some, believe it or not, very, very few, but I think some cancer cells can actually still use some fats through fatty acid oxidation. It's
Mhm.
very little, but so the ramifications of that fact are really none, but it's very little. So some of them still, but it goes to show some of them still function differently. So if they can still use some fatty acids, well, how much glutamine are they using then? Because glutamine is used in the Krebs cycle.
Mhm.
So how much, like, is a ketogenic therapy going to have the same associated results in one cancer population as opposed to another cancer population if those cancers are different? You got to be. It's just really important to be careful with that. So
Yeah.
if you want to make an index, that would be the best way to do that. What is the most associated index, plus or minus, to find an interval range again.
Mhm.
And I mean, go from there, really. But again, broader the better usually. I would just trust the process of a ketogenic diet. And then if that's not working, think about glutamine inhibitors or something like that. If that's not working, or if it's not working fast enough too, it's working but not fast enough, well, yeah, I would undergo some chemotherapy,
Mhm.
because obviously what's important is maintaining the life of the individual.
Mhm. Right. And something you said a little bit earlier about the possibility of cancer upregulating the generation of glucose. I
Mm-hmm.
vaguely recall hearing, and maybe I heard it from you actually, I'm not sure, that when in the last days of cancer patients' life, they waste away, and that could be indication of their muscles being cannibalized for glucose.
Uh-huh. and
Um,
and I'm quite certain that that's what it is, because
I see.
they they will actually, I'm pretty sure that when observed, if you observe their body composition, like through like a DEXA scan or something like that, or I don't know how it's done on cancerous patients, but or cancerous patients, um, they I believe that their fat content changes very little
Oh,
compared to their muscle content.
interesting.
I'm not saying that it doesn't change at all. I mean, their fat can also be used in different pathways and less efficient ones to create glucose, but muscle is more readily. I mean, it's one of the reasons why when you consume protein above a certain amount, you'll actually see an increase in glucose production. Um, Sometimes shifting the I to G ratio up, so you actually get into an anabolic state. Most of the time, I would actually venture to guess. Um, so amino acids themselves, they can either be introduced exogenously, or your muscles can be broken down to produce glucose too, for glucose demand. Now, I think right there is just a, I think that is proof, actually, because these patients usually are consuming a lot of glucose by the hospital food guidelines that they're abiding by, that their nurses and doctors are abiding by. They're providing them with food that is just teeming with glucose.
Right. Yeah.
Um, But despite that, they still waste away, which tells me that the cancer gets even more hungry. I mean, granted, does it mean that the cancerous cell can actually have an influence over increased glucose production beyond what otherwise would have been produced by the liver? Not necessarily, because what could be happening is the cancer is just getting bigger. So the
Ah,
demand is being met by the body. Yes, like like, how do I put this? Maybe the glucose production by the liver is still staying the same. It's still staying the same, but the cancer has gotten larger. And so therefore the food that is being administered to these patients is now no longer enough glucose. So now the liver is still providing a steady flow, but the cancer is now depending on it even more than usual because the exogenous glucose was used up faster.
Interesting.
I don't like it's all mechanistic speculation here. But I mean, at the end of the day, I'm still struggling. Like, isn't that the exact same thing as what I'm saying though? Which is that it does have an influence over the demand for like because it's not starving. Like, so yeah, it must have an influence.
Yeah,
It must have an influence.
in a way, it's a little bit disturbing to think that it might, because then you open up this question of whether you, if you do go into a ketogenic state, are you inducing the cancer to start cannibalizing your muscles?
Right. It is interesting to think about too. Yeah, yeah,
That'd
exactly.
be,
Which is why I mean, really, I really respect Thomas Seyfried for what he's doing, but I would love to talk to him directly about this. I'd love to speculate with him about this because I don't know if he's talked about it a lot about this possibility because I feel like to draw a call like okay, yes, we know that glucose is dependent on my cancer cells. From that logic alone, that doesn't mean stop consuming glucose. There's other logic we use to determine that. That doesn't mean stop consuming glucose because that assumes that if you just cut off the exogenous supply, there's no other supply for the glucose. When there is, there's like that's not that there is another supply. Also, it doesn't mean that glucose was actually the thing that caused the cancer. That's another thing that people. I think
Mhm.
there's a superficial carnivore crowd that kind of just well, cancer cells use glucose. What do you know? And it's like, well, yeah, but that doesn't that doesn't mean that glucose was the cause of that.
Right.
Could have been, but that just means no. Now the cell is so deranged. Maybe by meat and fat. I'm just thinking in the devil's advocate realm
Mhm.
that now it has to use a bunch of glucose, like it otherwise wouldn't have. Like if it were using glucose oxidatively, so in the mitochondria, because they were actually consuming carbohydrates, then it wouldn't have gotten to this level. Like who's to say? But that's devil's advocate realm. That's not
Right.
what seems to actually be happening. But this is why I did a video recently. Well, I say recently; it was a few months ago, and it was on this very topic because it was like a relatively mainstream science channel. They were actually spewing some good information, some objective information for the first time, and I was like, they even said they're like, okay, so everyone thought, okay, well, since glucose is being used by cancer cells, that must mean that you should stop consuming glucose. So they tried it, and it didn't really seem to work. Granted, those diets weren't carnivore, but well, that's not the theory was if you cut the glucose supply off, then they would die because they depend on glucose. Well, it turns out that glutamine is also involved. However, these diets were not replete with glutamine because they were like plant-based keto diets. So what's the like? What are we dealing with? Because
Yeah.
I mean, are we like? I'm as objective as possible with this stuff. It doesn't seem to be carnivore is the be all end all. At least in this mechanism is not the explanatory mechanism for the association between an adoption of a carnivore diet and the amelioration of cancer or the regression of cancer in many anecdotes that are reported by carnivores.
Mhm.
Because I've received many anecdotes like I had cancer, I went carnivore, my cancer's gone, and I'm
Uh-huh.
not denying those, but that mechanism doesn't seem to actually be the clear one to me. There's got to
Okay.
I think there's a more detailed explanation that has to do or a more intricate explanation or a nexial explanation, spiderweb again of of repair mechanisms that or supplies that you're providing the body with now that you're providing it with different nutrients and all that stuff that have influence over the sort of a jump starting of a cell back to a regular eukaryotic stable state
Mm.
from its prokaryotic sort of cancerous state um, that has not really been identified. I do not think this glucose thing is a I don't think it's a good explanation, which might shock some people, considering in my book I wrote all about how the prevailing mechanistic speculation regarding cancer amelioration and stuff like that was carbohydrates. Like I talked all about how carbohydrates are involved in the cancerous process in terms of how the cancer cells use them and why cancer cells use them. So glucose should maybe be cut out, right? But at the end of the day, I didn't say that it was the be all end all. What I said was, okay, well, maybe cut it out then, because that could be something. It could have influence.
Mhm.
It's not saying that it's a causal influence at all. Who is? I think more research needs to be done, and we may
Right.
never find the answer.
Uh,
Yeah.
yeah, I saw Seyfried's and others, their recent science paper where they had a few glioblastoma patients who were put on a ketogenic diet, and
Mm-hmm.
I think they used either 36 to 48 months as a survival to that period after
Right.
diagnosis. And it was pretty intriguing, but it's so small, such a small study. So I hope they can do larger
Yeah.
studies.
It's always a with those studies. I know a lot of people, including myself, we attack the small sample sizes and the lack of reproducibility of results and stuff like that. The main reason I attack them is actually because of the people saying that that is in any way something that shows anything of any utility. The fact that there's a lack of repeatability, the fact that there is low sample size in these studies, that's actually not the thing I'm really attacking, because I understand why. Money is a big issue when it comes to like the funding of these studies.
Mhm,
It takes a lot of funding, and people that are in charge of funding it have to deem it to be appropriate to do so. So it also depends on who's in charge,
mhm,
which then gets political and all that stuff, which we're very familiar with. Will these scientific journals publish the study whenever it's done if we actually get it done? That's just depends on who heads the journals. I mean, you never want to attack the overall agencies, I don't think, unless the agency just genuinely shouldn't exist, such as like the American Heart Association or any of these scientific journals. Like you don't want to say, well, the American Heart Association, they suck and always will. It's like, well, the agency is run by people. So if you change the administration, then they could actually become really good and responsible. It's not the institution itself, it's the people that run it. So you always have to be careful with that too, but that's why it will necessarily get political. Like, who's in charge of these scientific journals?
right.
Who's in charge of the publishing at this moment?
Yeah.
And so there's a lot of politics that goes on in the scientific realm, and people don't realize that. So yeah, conducting studies is a lot harder for than to do than people realize.
I imagine. Yeah.
Yeah,
Mathematicians have it good.
yeah, yeah. I think so. Yeah.
Okay, so I mean, from everything I've been learning so far, it seems like it's fair to say that it's a good aim to minimize glycation damage and therefore to reduce the amount of sugar consumption. The less is the less the better.
Hmm.
But one thing that I've always been puzzled about is I think you mentioned that with gluconeogenesis, it doesn't really reduce its rate of glucose production when you eat carbs. I mean, it does a little bit, but it doesn't counterbalance the amount that you
Hmm.
start. So why is that exactly? What is your explanation for that?
I've had a theory about this. I based this off of two things. First of all, before I get into the reasoning that I kind of come up with, there was a study that I saw that showed they showed that postprandially after glucose consumption. No. I'm remembering now. They didn't consume anything. What they did was they did a hyperinsulinemic clamp on patients on in a clinical setting where
Okay, I've,
they
I've heard of hyper clamp. I mean, I clamp, but I don't exactly know what it is. I
yeah
don't know
well
what it is. I just
there's
heard it used. It's a really good way to determine insulin levels.
yeah, basically, to simplify it, they raise these people's insulin levels without causing the glucose to go super low, because usually what you'll do is if you raise insulin you can get hypoglycemic,
Hmm.
and then you can die actually if you get too high. So they clamped it. I have to look it in more detail because there's different types of clamps. There's hyperglycemic clamps, I believe, or either way, what they showed was that if they kept their blood glucose level steady, but they found a way, they raised the insulin level. Hyperinsulinemia inhibited hepatic gluconeogenesis, so liver gluconeogenesis, by 20%.
Okay.
So the idea was, well, in order to actually get to the level of insulin, you have to consume lots of glucose. So the insulin is actually the major regulator of gluconeogenic activity on the opposing side, but it's not enough of a regulator to the point where the liver will completely shut off glucose production until that other glucose in the blood is cleared. And then, it'll slowly, as the glucose level in the blood drops from exogenous input, the endogenous input will come back up and up and up and up. It doesn't seem to do that. That was my first inference. The second one was whenever you see type 2 diabetics, despite the fact that they have hyperglycemia, their liver is pumping out a ton of glucose still, a lot of it. In fact, it's one of the main reasons that metformin, it's one of the major selling points of metformin. It doesn't just bring your glucose levels down in your blood, it also stops the liver from producing a lot of it. So it's like a double, I mean, it's double as good, right? Well, the theory that I've come up with for people that are not diabetic as to why the liver still pumps out lots of glucose despite hyperglycemia, hyperinsulinemia, whatever it is, because they both are obviously, they coincide for obvious reasons, is because as much as we have an ability to phenotypically adapt to a diet that is clearly not appropriate for us to be consuming, diets that involve spiking your glucose 3 times a day, as much as that's true, it does not seem that we are as flexible when it comes to our genotypic maximum adaptation. So, how do I put? Yeah, does that make sense when I say that?
Because we've put ourselves in a environment that is completely contrary to what we evolved to adaptively.
Yeah,
So our gluconeogenesis is actually designed for that environment which we used to live in.
exactly. Like every species has an ability to phenotypically adapt. There's always an interval range for presentation, but where those cutoffs are is set by your genotype. You can't you can't just infinitely adapt, right? Like that's not how that works.
Right,
Species will go extinct if there's an environmental change that's too drastic and stark.
right.
Like that's so there's always a genotypic limit, and I think that that's what I've come up with. There's such a pressure on our liver to maintain glucose levels without exogenous glucose. There's such a pressure on us that our efficiency for it is so high that whenever you spike your glucose, the liver, yes, responds. Like the glucose production goes down, but it goes down only 20%. In the people studied, I understand that there's always limitations, but
Mhm.
20%, I mean, that's a pretty low signal.
Right,
Like that's a pretty low amount. That's a fifth. You'd expect it at least like if you like 50%, maybe like 60%. I mean, it doesn't matter though, because it doesn't go below. I mean, it doesn't go above rather 50%. It doesn't do that at all. Um, and so my theory was that, and of course, I'm very careful to say it's a theory.
Mhm.
In type 2 diabetics, by the way, I made the distinction because in type 2 diabetics, I think that there's an additional factor, which may actually be, in their situation, the more significant one. If they're type 2 diabetics have stored lots of fat in not just their adipocytes, their actual fat cells, but their muscle, their organs,
Oh, uh-huh.
um, their liver, for example. And so the idea there is, well, insulin is supposed to bind on to the receptors and supposed to activate phosphoprotein phosphatases and all these other mechanisms that were down to eventually inhibition of glucose production. Maybe not complete inhibition. Everything's a fader in the body. That's how chemistry is. It's not an on-off switch, but nevertheless, it's supposed to inhibit it. But if those cells are so deranged and they're so maximally hypertrophied because of, stored fat, the insulin cannot, even if it binds onto the cell, it can't do its job as well. This is basically insulin resistance is the theory here, where it cannot do its job as well. And so it is going to pump out glucose to the same extent that it was before the glucose level was as high as it is in the blood, because now it can't respond as well. I have qualms a bit about the theory because,
Hmm.
I mean, maximum hypertrophy of fat cells, maximum hypertrophy of all cells. Every cell has a maximum degree to which it can actually hypertrophy. It can't get beyond a certain size or else it will burst. It'll leak upon impact. We actually see this a bit in overly fat people. They will leak. Their fat cells will actually exude compounds into the bloodstream from the cell, um, which can almost cause a catch-22 situation because then the body can become inflamed a bit
Hmm.
because there's a bunch of foreign compounds in the bloodstream. Um, Especially considering your fat cells store a bit of toxicity as well. If you're eating toxins, it does it in other animals, so why wouldn't it do it in us? But um, I don't know if that. I haven't seen enough definitive evidence to suggest that that's the case in diabetics. If anything, I think that the stored fat in their liver isn't causing insulin to not work. It might just be inhibiting insulin from having its intended effect, like I just said, through a Randall cycle situation where there's a bunch of fat in the cell to be used, but it's not being used quickly enough because you just keep eating and eating and eating and eating and eating and eating, that there's a back pressure chemically, not physically, but chemically, that is not allowing insulin to do its job. Either way, I mean, the consequence is the exact same. The consequence is the same, which is, okay, fat's in the cell, it's preventing insulin from doing its job in the liver. The liver's not going to respond to insulin like it otherwise would. So not only, you're not just going to only get a little bit of an inhibition of gluconeogenesis or glucose production, you're going to get even less than that because now insulin isn't even there to signal properly.
Interesting.
So that's another big part of the picture. So it depends on the person, depends on their presentation, whether they've got, you know, diabetes. I really really hesitate to ever say insulin resistance because I cannot stand
Right.
using that term because of how ambiguous it is and how perverted the definitions are as well. So
I mean,
yeah.
I've heard some people look at the small decrease in glucose production and gluconeogenesis as indication that actually our body needs more glucose.
Oh, um, it doesn't make sense to me because when you look at situations that involve that massive amount of glucose production from the liver and the hyperglycemic spike, you will see much of it being transmuted into fat at the liver to be stored. Sometimes you'll also see a hypoglycemic rebound effect, which is reactive hypoglycemia,
Mhm.
which is also indicative of the fat. I mean, if you become disordered in your presentation after spiking your glucose, that should tell you that you shouldn't have spiked your glucose. And so that means it was too much glucose. So I think that's a I think that's a really, really bad argument, frankly.
Mhm.
Um, I don't know. Who have you heard that from?
Well, it's actually a friend of mine who has thought about a lot about diet on his own,
Okay.
and I've had a debate with him. I actually wanted to get your take on it too, because my response was actually your first explanation, which is that we're in an evolutionary um, divergent environment.
Mm-hmm. Yeah.
Yeah. Um,
Yeah. No, I think that the evidence of reactive hypoglycemia, as well as the fact that most of that glucose can be observed to be transmuted into fat directly at the liver, then to be stored in situations like that, is unequivocal evidence that it's clearly glucose in excess because anything in excess will be transmuted to fat. It wouldn't, if it were actually required, it would be used. It wouldn't be transmuted to fat and then stored and then have that fat be stored later or have that fat be used later when you're in a fasted state.
mhm.
That's not,
Okay.
fat is necessarily a consequence above a certain amount, of course, because you always have to have some fat on you, but it's necessarily it is the primary form of mass stored in the body for later use. So yeah, no. I don't I think that's just unequivocal evidence of that being the reality.
Okay. So my next question is maybe basic physiology and um,
Mhm.
how does the body normally deal with glycation damage? Like if a protein gets glycated, what does the body do to that protein? What happens to that protein?
Yeah, so glycation is actually a multi-stage process, but the initial stage is glucose. It's funny that you mentioned this because it's going to be in the next lesson on the
Awesome.
program. Yeah, yeah, yeah.
Okay.
We're getting into glycolysis, and what's not usually taught in biochemistry, it's not ever taught, frankly, is like what is glucose? What is glycation? And like what is what is the chemistry of it? Like, is it polar? Is it non-polar? Is it So glucose is an aldohexose. So it is hexagonal in form almost always. Um, and it has an aldehyde group on its first sort of carbon. Um, and what happens though is that when you introduce glucose into an aqueous solution, like your blood, for example, 0.002 to 0.02% of it exists in its linear form. And it's linear because it's become unchained. So if you think of just a hexagonal shape, but you just unchain the hexagonal shape into a line.
Mhm. Yeah, and I
Yeah,
understand. Yep.
yeah. You've got you've got glucose existing in that form with an exposed aldehyde group now, because usually the glucose is contained in the cyclic form, or the aldehyde is contained in the cyclic form, the hexagonal shape. So it's not exposed to anything. It's not reactive. The linear form is reactive because the aldehyde group is really reactive.
Mhm.
Aldehydes are very reactive, in fact, particularly to amines and other, basically proteins themselves, the exteriors of proteins. And glucose, as much as it's a very small amount that exists in its linear form, this is the amount that causes glycation. This is the form that causes glycation. And we know that you only need a very little absolute amount to cause serious problems with with glycation damage. So glucose will form covalent bonds with proteins, and they're very strong bonds, and it deranges those proteins. You can basically think of it. I mean, a really good analogy. Usually, I don't like these analogies because oftentimes they're used because they're catchy, but as a consequence of being extremely simple, they've lost too much detail to the point of sometimes actually putting the wrong image in people's heads. But this one's quite different.
Okay.
They Glucose is sticky, right? Like it's a sticky. Well, the same glycation is the sticking to proteins. Basically, it sticks to proteins. The linear form will stick to proteins, and it deranges them. And depending on what is being glycated, depending on what is actually being you know attached to and deranged, this can actually cause direct tissue damage. Um, but also, again, depending on what tissue is being damaged, it will not only trigger damage, it will actually trigger an inflammatory response for other reasons. So inflammation is always the response to damage to tissues or a potential invading pathogen.
Mhm.
But if you derange a protein, the body can actually be triggered because of it perceiving the protein as foreign now because you've deranged it. So sometimes you could actually get an inflammatory response for twofold reasons, both reasons. Glucose will damage a protein, so it's damage to tissues that the body recognizes. But then if there's any that are floating around that are being deranged, then it can trigger PAMP and DAMP cell receptors to react, formulate to produce pro-inflammatory mediators, and then flood the body with more inflammatory compounds, which oftentimes, if it's in high enough amounts and it persists long enough, can actually contribute to more inflammation because of reactive oxygen species production, which also damage tissues like hydrogen peroxide, superoxide, all that. So
I see. Uh,
yeah.
so it's part of the general inflammatory response.
Hmm.
Okay. Um,
Yeah.
so my next question is, do all mitochondria in a cell behave in lockstep with each other, or do they act independently? Because understand you've described how muscle cells can have even 5,000 mitochondria in there. And actually, that's part of a sort of broader question about how homogeneous our bodies are and how homogeneous is cytoplasm, or is the cytoplasm in the cells, is it pretty much uniform? Or are there different regions in the cytoplasm that are very different from others? And also, like the body, the environments that our cells find themselves in within our body, I guess there can be sometimes regions of inflammation and others that are free of inflammation. And where are these, what controls the sizes of these different regions, and um, how heterogeneous can it be?
Yeah. So I think a good way of answering this, inferring this rather, whenever you're observing people that are exercising, they do something called respiratory gas analysis. PhDs are actually they have to be involved in this process, but they'll um, they'll observe what's called a respiratory exchange ratio, which the pedant in me doesn't like the fact that it's called a ratio because a ratio is something to something else or a fraction or something, but it's a quotient that they report. But whatever, it's the same sort of concept. They what they do, I don't know the exact specifics of the values, um, but they're effectively measuring the amount of there's a ratio. It's carbon dioxide is involved in that ratio, and I cannot remember. I think it's a carbon dioxide to oxygen ratio. It might be, which would make sense because we breathe out carbon dioxide, some oxygen, sure, that isn't used, but when you're exercising really intensely, it gets lower. And then the oxygen is consumed through inhalation. But what they do is they'll slowly over time see that as you perform the exercise, the ratio will start to change from a ratio that indicates fatty acid oxidation being the primary pathway that's providing ATP, producing it in mitochondria, to pyruvate oxidation
Hmm.
because you're engaging in really high intensity exercise, which is to be expected because you get such a large amount of pyruvate building up from muscle glycogenolysis that it's too much for the body to oxidize at that time, but eventually the equilibrium will shift and it will start to oxidize more. Um, but the reason I bring this up is because every single thing in the body is a fader. So what this tells us is that you don't just go from beta oxidation being a systemic pathway to glycolysis or what's considered aerobic glycolysis. I'd like to rename that to pyruvate oxidation. You don't go from beta oxidation to pyruvate oxidation like flipping a switch. It's always a slow shift towards that, which tells you that cell by cell, every cell responds differently. It's a systemic shift. It's a slow and steady shift. It's a fader being shifted one way or the other. Now, the question then becomes okay, well, if that's the case for cells, sure. But what about for the mitochondria in the cells? That's really what you're getting at. Like
Yeah,
within each one cell, are the mitochondria all doing the same thing? I think in theory, I don't really have a definitive answer for this, but I think in theory they could. But I think you'll struggle I think you'll struggle to find an example of that really occurring in a cell because usually they are functioning like it might be also a fader change as well because it's all chemistry, but the change between off to on if it's a fader, right? So dim to bright instead. Um, the change between those two endpoints could be faster with respect to mitochondria than it is with respect to a cell basis, than respect to a systemic basis.
okay.
Um, but I don't really have a definitive answer for that. I believe in theory that yes, that's correct because I don't really know how you could, if you have multiple mitochondria in a cell, in theory one of them could be damaged and the others couldn't. It could also not be. But the one that's damaged is not going to perform the same way as the others. They're all
Um,
individual organelles. So I would struggle to think they don't function in lockstep. They're not necessarily. This goes back to my point. They're not necessarily operating lockstep. There's nothing that actually indicates that they have to.
Okay.
But you'll struggle to find an example of them not, because they're all within the same cell and they're so small compared to a cell that any cell that receives a signal to sort of adopt different behavior, the mitochondria will respond. That signal is going to be a lot stronger to mitochondria than it's going to be to the overall cell, if that makes sense, because of the size difference
Okay.
and so the
And
concentration
that's
difference.
actually one of the things that I'm wondering about is the relative sizes of all these things and how far mitochondria are spaced. And but I don't even know if these things are important to think about.
Oh well, I can tell you right now that one cell, it's going to depend on the type of cell for sure, but usually it's measured in diameter in micrometers, I believe.
Okay.
um, and one cell, one average size cell, I suppose you could say, I think is about the size of the of a head of a pin,
Okay.
and the mitochondria are about a tenth of the size
Oh,
in terms
wow.
of diameter, I believe. Yeah, yeah,
But
I believe
then
so.
for muscle cells, this muscle cells must be much bigger.
Yeah, they're one of the. Well, I learned this a long time ago. Muscle cells are the the largest cells by mass and volume, so also then consequently by density too. The densest cells, they're the most amount of mass per cell. They're the most amount of volume per cell.
Oh, wow.
Um, and the only thing that comes close to even competing with muscle cells, particularly skeletal muscle cells, by the way, um, is uh is nerve cells, but not because of mass or volume or density, but because of length.
Oh,
Um,
interesting.
so skeletal muscle cells are also longer, but nerve cells are the longest cells in the body. Like you can have a nerve cell that's up to a meter long, I believe.
Wow. Okay.
Yeah, yeah. I think there's a nerve cell that runs from the low back to the foot.
Oh my God.
Yeah. So we're talking about like very long cells. Yeah.
That's incredible. I wonder how they discovered that. How did they know that that was the same cell? I
Yeah,
mean,
I know. I mean, you'd have to trace the whole thing down,
oh my God.
to clearly through an autopsy situation or something. But yeah, no, it is very interesting to think
That's
about.
amazing.
Yeah, yeah. Yeah. It's also interesting to think about how all the cells connect to each other too. Like they're all of them are connected in some way. It's very cool. That's whenever you get into more cellular biology and not biochemistry, where you actually look at the apical membrane of cells and then the epithelial cells and then like or the what's it called, the basement membrane, and like how all the cells interact, like they connect to each other, the cell junctions. You learn about the interstitial fluid as well as a consequence of cells being sort of stacked on top of each other, integrated through many different cell junctions, some of which are actually involved in cell communication. So not only do they hold cells together, some of them allow for the flow of information effectively through cells all throughout a part of the body.
Oh, wow.
Yeah.
Yeah, I mean, and actually, that's another reason why I was asking this question because I guess I understand that continuous glucose monitors actually monitor the interstitial glucose
Hmm.
levels and how homogeneous is that? So if you move the monitor sensor an inch down, are you like in another world in your body where,
Yeah.
or is
Think
it, yeah,
Yeah, things as broad as like glucose and that are carried by blood and are really that are so fundamental for a human system. Typically, it's quite homogeneous in
okay,
that respect. But again, it still exists as a fader, and it also depends on each cell's individual need for glucose, for oxygen, even like if we're gonna shift it to some other fundamental compound. Um, there's an element of both there, but in a normal situation where there's no ischemia, there's no um, blockages in blood vessels, for example, which those oftentimes go hand in hand, because obviously ischemia happens when you have low oxygen supply to tissues. So um, then they're quite homogeneous. So if you moved it to another interstitial fluid or another part of the body to measure interstitial fluid there, you may very well expect the glucose level to effectively be the same. Um, but don't quote me on that because there could be some nuance there. Believe it or not, as much as I really enjoy looking into this stuff and as much as I've looked into it for years and know a lot about it, there's also, just like you said at the beginning, just like we talked about in the beginning, there's so much we don't know. And
Yeah.
and and by we, really, sometimes I do just mean me. Like sometimes we collectively do know that, and I just haven't looked into it yet. So
I see.
yeah, yeah.
Well, um, yeah, I mean, I kind of wish that LLMs could be a solution to by being able to read all the literature and providing us with, accurate information of all the scientific literature, but unfortunately not.
Yeah. I argued with. Well, I uploaded a video on the early access
Yeah, I
program
saw
talking
that.
about
I
how
saw that.
I argued with ChatGPT for like 2 hours straight about the whole acid-base balance thing, and it was just telling me. I think did I explain why I was arguing with it or
I
like what
don't
it said?
I don't remember. This was a few weeks back. I do remember though that the video you made where you were asking, I don't know if it's ChatGPT or some other LLM, but
Yeah, I was arguing with ChatGPT trying to get it to I've been arguing for a year with it about acid-base
oh, I
balance,
see.
particularly I've got it to break on basically every subject matter, every single one, except this one, the
Oh, interesting.
proton. Yeah, this proton pumping phenomenon, and we'll get to that in the course. Like how the mitochondria actually produce ATP.
Oh, I can't
The,
wait. Can't wait.
yeah, the idea is that just to give you a little teaser, whenever you produce this compound called you produce NADH and then FADH2, but the primary one is NADH that you produce. It's an electron carrier,
Hmm.
and it'll supply its electron. So it'll break apart into NAD positive, and then it'll supply an electron into a system called the electron transport chain on the
Hmm.
inner mitochondrial membrane. And it'll
Hmm.
go to complex 1. And so complex, you just think of them as like little tubes, I guess, for like ease of conception.
Hmm.
And they're designed to be little like pellet guns, I suppose, where they build up a lot of energy because the electron travels through these what are they called?
I think I've read a little bit like the iron sulfide
Iron and sulfur cluster proteins.
cluster
But there's a
proteins. Yeah.
they're specifically called something I can't remember. But they go, really, but it builds up energy, and then the complex uses that energy to pump a proton H plus
Hmm.
into the intermembrane space. So you've learned the beta oxidation lesson, so you know the sort of the very simplified, and this is on purpose, the very simplified sort of conceptual image of the mitochondria. You've got the outer layer,
Hmm.
and then you've got the inner layer, and that forms a matrix and an intermembrane space.
Right.
So it pumps a proton into the intermembrane space. That's the idea. That's the idea.
Yeah. I just uncannily, my next question was going to be about this exact
Okay,
topic.
good. No, ask it. No, I can cover it. I can cover
Because
it.
I was
Yeah.
wondering, what guardrails are there on this electron transport chain ladder that prevents NADH from just reducing oxygen straight to water? How does it stay on this ladder? Um,
Um,
I mean, if
I believe.
oxygen's all around, everything's all around at once, and
Uh
oxygen wants
oh,
electrons
that's
so badly, why wouldn't it
oxygen.
just take them?
Well, oxygen is only, from my understanding, oxygen is only in one part of the mitochondria.
Oh, okay, interesting.
Yeah, it
Okay.
stays in one part. Yeah. And it also, by the way, doesn't exist as O2. It exists as O1.
Oh, okay.
Yeah, it exists as atomic oxygen, which is hardly ever seen in the universe in terms of like actual like exposure to oxygen. When we observe it, it's almost always an O2 because it wants to form an octet, chemically speaking, with its electrons. And
Yeah.
so it'll always bind to itself usually. It doesn't make it less reactive. It still makes it very reactive, which is why things oxidize. They rust, things go bad, spoil food, you know.
Mhm.
But that somehow, I don't know the process, I really don't know the process, but somehow our body separates that. They separate the oxygen molecules and then exists as atomic oxygen in our mitochondria, in our mitochondria.
Wow.
So yeah, yeah. But that's why. So if you can imagine, like chemistry, I always think of like magnets, basically. Like if you have oxygen in one spot, then obviously the NADH will want to just pull. It'll be attracted to one spot instead of it being all over the mitochondria. Yeah, oxygen is in specifically one part of the mitochondria.
Oh,
I'm almost certain of that fact, and it's always at the end of complex four. Um, I believe called cytochrome C oxidase. I think, which would make sense because becomes, you turn it into water.
I
Um,
really have to, because I was kind of imagining the inner membrane of mitochondria as being just studded with complex one, two, three, and four,
mhm.
in abundance everywhere, sort of. So, like everything you
Yeah,
need everywhere.
yeah, yeah. I suppose that makes sense too because yeah, there's not just one electron transport chain in a mitochondrion, right? I mean, there's also multiple. There are multiple ATP synthase proteins in the mitochondria. There's multiple, and there's also some that are like H0, H1 ATPase. There's U0, U1 ATPase that responds to UV light
Oh,
exposure.
interesting. Wow.
Uh
Okay.
huh. So that's one of the ways that sunlight can actually increase the rate of mitochondrial function is by interacting with that protein.
Oh, wow.
So you can actually get an increase in the rate of metabolism by just being out in the sun.
Amazing.
Yeah. So I don't. I'm. That's a good question. I should definitely look into that because yeah. I mean, if that were the case, then of course that's a good question. There's got to be some strong chemical pressure for NADH to go to complex 1 first, and of course FADH2 to go to complex 2 first, because that bypasses complex 1. Clearly, it has to do with chemical structure. But your question isn't why they go to those first. The question is how the electron stays on course through the chain.
Yeah.
Yeah. So
And it would have to for those proton pumps to work, right? Because,
you do. I mean, I will say, you've got a CoQ enzyme that is a mobile transport enzyme. I almost want to say protein, but it's the only enzyme in the body that isn't made up of protein. Really interesting. Um, but it does move protons between complexes. But the question still remains as to how, when it gets to complex three, why doesn't it just react with oxygen right there instead of going to complex four
yeah.
and then going to, well, actually, not complex four. There's complex three, and then I believe there's cytochrome C, and then there's cytochrome C oxidase, which is complex four. I think there's actually two more after complex three. I have to review a lot of stuff. I'm gonna have to review first, do a cursory review before I do lessons. But um, yeah, no, that is an interesting question. I'm not sure. I'm not sure. I mean,
Okay.
either way, I do believe the oxygen, if it's not in one part of a mitochondrion, because there's multiple electron transport chains in each mitochondrion, you've got to have you've got to have almost like pockets of oxygen, and it's not just a free floating according to its concentration gradient kind of thing throughout
Amazing.
the mitochondria. It would almost, I feel like it would almost have to be that way. It almost have to be that way. I can look into that. I can look into that because all that detail I think would be really cool to include into the course.
Yeah, I would love that. I love it. Actually, I totally was lapping up everything you were saying about beta oxidation. That was all new to me. And I was just, yeah, that was really cool.
Yeah, 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 yeah. No, it is really cool to learn these things. It is. Yeah, what I was saying about proton pumping, just to finalize though, is that yes, the electron goes through, but each time there's some complexes that are not involved in proton pumping, but especially complex one is that's typically the one it's the start of the chain. So complex one and then complex three. Complex one, what's said is that the electron goes through and it causes a bunch of energy that causes the complex to pump a proton into the intramembrane space from the matrix. So you've got a bunch of H plus in the matrix, and then you've got a bunch of H plus in the intermembrane space. And then the idea is that you pump a bunch of protons into the intermembrane space, and it forms a concentrate according to its concentration gradient. They flow back in to the matrix. It's just a cycle, but it flows back in through ATP synthase. So as it flows in, it spins a motor, and that's what causes ATP to be synthesized. To really simplify, it's like that's what's going on. So they say it's a concentration gradient, right? That's what's causing H plus to flow through these fluid pools between two fluid pools. It has been established. I don't know why they are not. I don't know why this is even posited as being what actually happens. I have no idea. This is one of the most prevailing theories, yet they admit that it's a theory. It's called the chemiosmotic theory. It's how the body produces ATP through oxidative phosphorylation, the mitochondria. It's called a theory, but they pretend like this is just a fact. This is how it does it. It is a scientific fact, and this was proven decades ago. You cannot transfer H plus between fluid pools because they're not independent variables on their own. You can't do that. So it's a literal physical impossibility. You can't adjust the concentration, for example, of H plus in a solution by manipulating H plus. It's a dependent variable, and it's dependent on 4 factors. 4. That's what pH is. It's the concentration of H plus in a solution, quote unquote.
Right.
You can't adjust pH.
Oh.
You can't adjust pH by changing directly the concentration of H plus. You can't do that. It's not possible. It's physically impossible. Like that, I'm very, 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 very rarely am I convicting on things, believe it or not. But that is something that is a physical. I've argued with ChatGPT about this for a year. It admits it. It admits that you can't do that. But then it'll simultaneously say, well, yeah, H plus is pumped through the inner mitochondrial membrane between the matrix and the intermembrane space. And I go, well, that's not. You can't adjust the pH of a solution by adding H plus to it. Just like you can't do that by removing it. So I'm not denying that there's a pH change between the solutions when there's electrons being pumped or electrons being administered into the electron transport chain and are going through those complexes. But there is no physical pumping of H plus. And I argued with it for 2 hours a few days ago because I was trying to tell it, like, I was trying to argue with it to admit the point. But at the very end, I said, okay, tell me if this has actually been observed before. I said, finally, I was like, just tell me if this has been observed. And of course, first it says yes, and it gives me the studies and tells me exactly how it was done. I said, one of them said, well, what it showed was that there were changes in pH according to pH fluids that they administered, like pH drops, where you can adjust, you can see the change in pH according to the color of the fluid between the matrix and the intermembrane space. I said, that's not right. That doesn't prove that protons are being pumped. It proves there's a change in pH. So tell me, has there actually been an observed proton pumping phenomenon?
Oh.
And it said, no.
Oh, wow.
It's never been observed. You can't have a concentration gradient of H plus that forces protons into the matrix from the intermembrane space, like a buildup of something, so then it just needs to flow back in through a motor or something like that, like ATP synthase. That's not how H plus works. It'll build up energy. It will, but it won't. You can't adjust the concentration of H plus by manipulating H plus. I'm not pretending there isn't a change in pH, but you can't say that. That is not physically possible. The only time that would be physically possible is if that's not actually what's happening, and what's happening is the two fluid pools become one for an instant. But even then, if a fluid pool becomes one, it becomes one singular pH.
Right.
One singular pH. If I took a glass of water and then I just put it into 2 different glasses, they'd be the same pH then. Like, that's not. Once they become one fluid, they adjust in pH almost instantly, effectively instantly, less than 1/millionth of a second. And then if they tried to make them 2 fluid pools again, intermembrane space and matrix, they'd be the same pH. But that's not what happens, because they're always different pHs from each other. Always. So something's going on, and we haven't figured it out.
Oh, wow.
We have not figured it out. Something's going on. I'm highly confident, but of course, always want to exhibit humility. I'm not like convicting about it. One of the four variables of pH is always one of the most common ones that's actually the cause of a change in pH, and it's called the SID value. Also, Stewart, the pioneer of this model of pH balance, acid-base balance, he called it the SID plus value because in biological aqueous solutions, it's always a positive value. It's it's the strong ion difference. So it's the difference between it's effectively the net charge of a fluid pool. Um, Theoretically, the net charge of a fluid pool always is zero according to the law of electroneutrality, but it's the amount of ions, like negative to positive ions, in a solution besides H plus.
Okay.
So basically, if you have a solution and you put a positive ion into the solution, even small changes, extremely small changes like that, can cause massive discharges of current, charge differentials.
Okay.
Can cause massive discharges of current. So if what I'm saying is that if the law of electroneutrality were not universal, if you put a positive ion into a solution, that thing would explode, like instantly explode. Same thing if you put a negative ion in the solution. But what happens is something adjusts in the solution. That's pH. To prevent, as per the theory, an explosion. It actually has a charge equilibrium that occurs in a fluid to prevent spontaneous explosions of fluids. That's why when you consume salt, you don't explode. Like that's not, there's something that adjusts. And what it is it's pH. And the theory goes that according to water dissociation, so you've got H2O, but H2O could dissociate and does dissociate into H3O plus and OH minus in a solution. There's a certain rate at which it does that, and it's dependent on, well, these four variables, because that's what causes pH. H3O plus is effectively H plus. H plus never exists as H plus in a solution.
Okay.
It's a stoichiometric.
Interesting. I
Um,
didn't know that either.
yeah, yeah. H
Yeah.
plus never exists as H plus in a solution. You'll often see that people will say pH, right? It's like
Right.
I'm using it in a simplistic way. Really, pH is pH3O plus.
I see.
It's the potential of H3O plus. So the concentration. This is a really dense topic, really, really dense. But that's why I'm really excited to talk about it. Um, but it's according to water dissociation. There's four factors that regulate this.
Well, but if you could say quickly, what are those four factors?
Yeah, yeah. Well, that's what I'm about to say. You
Oh,
got four
okay.
factors.
Okay.
SID plus. Again, really, it's SID if you're going to talk about general solutions, like outside of the context of biological solutions. But in the context of biological solutions, it would be SID plus because it's always positive, and I'll explain why. Then um, you've got ATOT concentration, which is the concentration of weakly dissociated acid anions, and I'll get to that as well. Then you've got temperature. So if you heat water up, it will become more acidic. If you cool it down, it will become less acidic. And carbon dioxide concentration in water. So water will act as a solvent for carbon dioxide in the atmosphere. So there will always be some carbon dioxide gas in water. But as a result of that, it becomes slightly more acidic if you were to take water out of a vacuum, a theoretical vacuum, and expose it to the atmosphere.
I see.
So normal, like neutral pH of water, in the way that it's conceived to be neutral,
Hmm.
is actually 6.8 roughly,
I see.
instead of 7.0. Um, so the thing that happens, and I can't get into all of pH balance because that would be, I mean, that's a story that's got to be in the course. But
Okay. Okay.
the the thing I was getting at was that SID is the most common influencer of pH because what happens is a positive ion. Again, if you introduce a positive ion and nothing regulates that charge, nothing negates the charge, the fluid pool would explode. So what happens is water dissociates. Or rather, no, less of it dissociates. More of it will rejoin to form H2O to prevent the charge. You've got H3O plus in the solution. If you put another positive charge in there, you've added a plus one. Now it needs to negate it with removing a positive, or at least adding a negative. So water rejoins H3O plus and OH minus reform to form water, as per the theory. The crazy part is actually we have no idea what is actually negating the charge. That is the theory, the water dissociation theory, which was actually disproven. We have no idea. The model still works. We use the water dissociation as an accountancy sort of tool and a conceptual model of it, but it's actually not what's going on. We have no idea what's going on, but that is what happens.
Wow.
Positive charges have an alkalinizing influence over a solution, because if there's less H3O plus in a solution, then there's a higher pH because it's a logarithmic function,
Right.
actually. So it has an alkalinizing solution. Negative ions have an acidifying solution. So what I think is going on is if there's a change in pH, we're talking about electrons that have negative charges going through an electron transport chain. I believe as a consequence of it traveling through and interacting with the surrounding fluids, potentially, there's a change in pH going on as a result of that. I think that that's probably what's going on. It could also be a consequence of not really even the electron, but NADH is neutral, but when it relinquishes the electron to the electron transport chain, there's an NAD positive left behind. So pH in the matrix will change. Particularly, it will rise, even if slightly. And the intermembrane space, though, that's not explained why the pH changes there, because nothing is actually being introduced into the solution. They keep saying that H plus is introduced into the solution, but it's not that's not true, because you can't do that. It's not a physical possibility. Like I said, ChatGPT will admit that. It'll say, well, yeah, that's not possible. And then you'll go, okay, so then how is it pumping protons? And it goes, well, because it is. Like, it doesn't actually answer the question because it doesn't know how to. It can't actually think, you know.
Right.
So it's a mystery, and it's one that has intrigued me for a very long time. Actually, I don't know what's going on.
That is amazing. That is very intriguing.
Yeah.
Wow. I'm now going to be haunted by this, because this seems just too fundamental to have a whole of in our knowledge about.
Well, when I learned that the chemiosmosis theory was called a theory in the first place, I was so confused, because this was years ago, and I went, "Oh, it's a theory? What? Okay, that's interesting." Which means that it has never been like actually materially, tangibly proven, if that makes sense.
Yeah. Wow.
It's just a theory, and then you get into this, and you're like, "Oh, now it makes sense why it would be a theory, because it's literally based on an impossibility of physics." They don't realize that, because there's many acid-based models that actually do believe that H plus, they treat H plus, they've been disproven. I'm sorry. They can, on a broader level, they can work, but like when you actually get into the nitty gritty, they don't work. Even this one is no exception. It's just the most robust one. There's some that treat H plus as an independent variable, but it's not that is not physically possible. You cannot have that be an independent variable. They're never independent variables under any circumstances ever, unless you were to like launch plasma into a. I don't even know how you do that. That's I don't know. I don't know. But yeah. So whenever people like, have you ever heard of proton pump inhibitors?
Yes, I have.
Yeah, they don't inhibit proton pumping. It's not what they do. They what they do is they inhibit chloride ion pumping into the fluid in your stomach, which is actually what acidifies the solution, because you've put Cl minus into the solution, which causes more H3O plus to dissociate from water in the solution. It acidifies your stomach by pumping chloride ions into the fluid. They're not pumping H plus into the solution.
I see. Ooh.
Yeah.
Um,
It's really, really interesting.
so
Yeah.
when you do this module for the biochemistry class, I really hope you make it clear what is definitely known.
Yeah. No, I will. I
'Cause
actually put a note on there that says to cover the epistemological
that might
undergirding.
okay, that's great, because I mean that request might just be a plea for me right now, because I am beginning to lose a sense of that here in on this topic. Um,
Like in terms of how we know that something is true.
well, for example, has it been established that the pH levels are different in the mitochondrial matrix and the inner membrane space? Is that definitely
Yeah.
okay? So that is, and the electron transport chain itself. I mean, this whole thing with electrons being shuttled about on CoQ10 through these iron sulfide
Yeah, iron sulfur
that
cluster proteins.
sulfur
Yeah.
close. Yeah, those things. Is that a well-established fact?
Um, I believe so because it does actually match within the confines of physics and physical reality. However, I mean, technically, if you want to be responsible and question everything, I haven't actually seen the original papers that show that and any future ones that have been done on the topic.
Okay,
To be extremely fair, yes,
has it been
that's
observed?
yeah, I believe so. I mean, we do. We also know that drugs that act on that pathway. Well, I guess this wouldn't be a good argument because proton pump inhibitors are based on the idea that you pump protons, and they also have their intended effect, but they don't. They're not based on reality. So, um,
It's
has it been
interesting
observed?
because I
Yeah.
like of course of late I have been looking at all these animations of the electron transport chain on YouTube
Uh
to,
huh.
to just get a visualization of it. And it's true that they only get to a certain level of detail when it comes to the pump itself. And it'll be like a cylinder, and then they'll
Uh huh.
show this H plus thing going through it. But I've always been like wondering, how is that actually happening? I
Yeah.
did read one source which said that there was a protein that had an H plus on one side and then it got rotated.
Yeah. That's ATP synthase. I believe. I believe you're talking
Oh,
about.
no, I'm
Um,
talking about in the complex 1, 3, and 4, that
yes,
that's how
yes,
the
yes,
H plus
yes.
gets put into the intra membrane space.
Yeah. What? Yeah, I just now remembered. The only thing that has actually been proven with respect to proton pumping is that there are proton binding sites on these these complexes and on other parts of cells,
Okay.
which means that yes, H plus has an ability to interact with the complex. It doesn't mean that protons are being pumped. They'll
Is
often.
there a protein that's rotating? Was that
I was never taught that.
okay? Okay.
Not that I remember.
Ooh, this is disturbing for me because I've always sort of put cell physiology and biochemistry as an anchor from which we can rely on. And now this is the first time I think for me that I've encountered something where it's a bit dubious.
Yeah, whenever you get to the granular details of like mitochondrial function, yeah, that's whenever it starts to go awry. Now, fortunately, that's you're already pretty close to bedrock whenever you get to that point to where it's like, okay, like I'm less concerned about it there than I am when it comes to like the problems that arise even just at the top levels when you investigate of like statistics and like the reporting of statistics and stuff. It's like, oh my God, you got all this up here wrong. I can't even imagine what's going to happen when I dig even deeper. But if you're already like right here,
I see.
it's less significant to me, at least personally. But it does, because at the end of the day, the model, even if it's not based in factual reality, the model works well enough to explain what's going on, and it clearly works well enough for drugs that are intended to have an effect on this model of mitochondrial function
Oh,
to have its intended effects, which it does say something. Um, it does. I mean, that's pretty significant. It's tough to ignore and shouldn't be ignored. But that's sort of like saying, well, I mean, it's less. It's actually less egregious. My friend, the coffee study that I was telling you about that I reacted to,
mhm.
or the caffeine thing. My friend was the one who sent it to me, the study, because he's in statistics right now. He started college, so he's younger than me, but he started college and took a class and he's my editor. And he said, "Oh boy, this will be real fun. This will be real fun." And he showed me an assignment that they were given, and they had to comment. They'd like comment on the assignment and like what it says and all that. And I said, "Really? Okay. Um, show me the study." And I went, "Oh my, ah." So I said, "Wouldn't it be funny if I reacted to that and then you could show your professor?"
Oh,
And he said, "Oh my God, dude, that'd be so great." So I actually recorded it, but I mean, obviously I was going to post it publicly, but you'll see that, the video, if you listen to me talk, it's like I'm talking to a classroom of statistics students because it was actually recorded for his class. I wanted him to like convince his professor to show it to the class. First, she has to watch it herself, and she's ready for it. Apparently, she's ready for it to
oh, that's
upload and
great.
stuff. So yeah,
Oh, that's
yeah,
pretty exciting. Wow.
yeah, yeah. But okay. Uh, where was I in explaining this? I always lose
Well,
my train of thought. Um,
I think you actually did explain what I was asking about the guardrails, electron transport chain. So if you wanted to wrap it up now, I would totally understand.
well,
And
I know that I was in the middle of something, and I can't remember exactly what I was at the end of it, and then I was gonna wrap up, but I can't remember exactly what it was. Um,
um,
something along the lines of, yeah, we talked about electron transport chain, and then the SID values probably what changes it
yeah.
seems or something. It's less significant. Oh yes, yes. It's like it's sort of like saying, and again, this is a more egregious example than the example I'm giving, or that we're talking about, but it still follows the same sort of pattern, even if it's on an inflated scale. It's like saying the calories in, calories out model.
Mm.
Like the whole calories, you consume calories, it's calories in, calories out, thermodynamics, first law of thermodynamics. It's like saying that since overeating food can make you fat and undereating food can make you skinny, that calories in, calories out must be true.
Oh, right.
It's like, well, okay, broadly, on a very broad level, okay, then that's true, but doesn't really have to do with calories. It's the same thing with the mitochondria. Like, oh yeah, these proton pump inhibitors have their intended effects. They work, so it must be. Yeah, but is it because of proton pumping? Is it because of that, or is there a slight nuance there that needs to be covered? Um, and I think it's again the calorie thing is a more egregious example of it because it doesn't work half the time because people like that's not it only works on the extremes, but it's still the same sort of philosophy if that makes sense.
Yeah, I hear what you're
Okay,
saying.
yeah. Well, yeah, we've been going for a while, and I've enjoyed every single minute of it. But I do have some things to do, and I have to catch up with people in the school group as to when I'm going to be scheduling this. I think it'll be tomorrow morning. So hope to see you there. I think you said that you'd be able to be there, correct?
Yes, that's right.
Okay. Okay. Good. Looking forward to that. And yeah, no, Ken, it was great talking to you.
Oh, thank you
Um,
so much, Eddie. I really appreciate your time. It was great for me. I really appreciate all your detailed explanations. They're fabulous. They're great.
awesome. Yeah. No, I'm glad that you enjoyed. And like I said, I enjoy these conversations too because they're intellectually stimulating, and that's important for my profession, if you want to call it a profession. Um, but yeah. Well, look, feel free to schedule another one with me as soon as we are both having a convenient time to do so, and as soon as you want to talk again, which may very well be directly after we end this. I relate to the experience. So, but yeah, totally fine. Email me, and or get a hold of Coach Peter about that, and then we'll go from there.
Okay,
So,
thank you so much, Eddie. Thank you.
yep. See you soon.
Yeah.